Okay, so in this lecture, what we're going to do is we're going to look at skeletal muscle structure, and then we're going to again go through step by step. How does a muscle cell, how does a muscle fiber contract? Now, as I mentioned in class before, when I use the term muscle cell and muscle fiber, it's the same word, same term, it's the same thing. I'll use those interchangeably. And so I want to start here with the model we used in class. And this is inside the muscle cell. In fact, here we have the different microfilaments and different structures inside the cell. You're going to go through a lot of this in lab, and so I'm not going to touch a whole lot on this here, but a lot of your lab model, your muscle cell model, will have T-tubules, the sarcoplasmic reticulum. You even have the myosin and actin. You'll be able to see A bands and I bands. A bands is where there's just myosin and actin. I band is just actin. H band is where there's just myosin. As that's where we want to start, because everything we're going to talk about today starts inside the muscle cell. As we talked about previously, we have a combination of actin and myosin. And in class, what I talked about is how they overlap each other. So we have actin, actin, the thin filament, and then you have myosin, the thick filament, and one kind of rests upon the other. And as we'll see, there's a step-by-step process where inside this muscle cell, thousands upon thousands of these filaments, actin and myosin, will begin to move along each other, causing the entire muscle cell to contract. Now, one term I want to mention here, just so we're clear on it, when we talk about the muscle cell, we often use the word sarcomere. And sarcomere, literally from the Latin, means measure of muscle. Measure, mere, sarco, muscle. So when we talk about sarcomere, we're talking about this area right here between a Z line and a Z line, which really is just, to make it really simple, it's just an area where you have actin on one side, actin on another, and myosin in the middle, and it's a measure of where these two would contract. Okay? To give you an idea, inside a muscle cell, you would have a sarcomere here, another one here, another one here. They literally would be lined up back to back to back, tens of thousands of them. And so we're just going to look at one sarcomere, because what happens in one sarcomere happens in all of them. And so if one sarcomere contracts, they all contract. And so let's kind of go through the process here. And again, this is another picture of what this looks like, but I want to go through an animation of it, so it just makes it a little more clear for us. So I talked about these steps in class. As far as these steps, I want you to know as far as the steps in muscle contraction. So you can look at this on your slides. It's on there on Canvas. You can take a look at that. But we're going to look at an animation of this right now. So let's pull this up, and here we go.
In order to produce movement in the body, the muscular, nervous, and skeletal systems interact in a process called muscle contraction. Muscle contraction begins when the nervous system generates a signal. A motor neuron conducts an action potential to its link with a muscle fiber. At the neuromuscular junction, a neurotransmitter called acetylcholine is released. This chemical binds to receptors on the muscle fiber. Acetylcholine receptors are chemically gated ion channels, and when acetylcholine binds, they open, allowing influx of sodium ions into the muscle fiber. This stimulates the sarcolemma and generates another action potential that travels through tiny folds known as transverse tubules. The action potential reaches
Now these transverse tubules, these are what we call T-tubules as well. And so understand here what we have is a neuron. Action potential travels down it to the motor end plate. Acetylcholine is released, interacts with the receptors on the muscle cell membrane, causing sodium to come in. And now this message is going to travel down these two tubules to literally every sarcomere in the muscle cell.
Fibrils within the muscle fiber. Surrounding the myofibrils are membranous sacs called the sarcoplasmic reticulum. The arrival of the action potential triggers the release of calcium ions stored in the sarcoplasmic reticulum. They diffuse into the muscle fiber, and the next phase of muscle contraction begins.
Okay, so this is where, again, calcium is going to enter into the cell, and you will see this next part here as it starts to bind.
When an action potential travels to a muscle fiber, a series of steps begin within the fiber that result in contraction. Muscle fibers are organized into units called sarcomeres that contain two types of filaments. Thick filaments consist of myosin. Thin filaments consist of actin. Eventually, these two filaments will come together, creating a cross bridge. In response to an action potential, the sarcoplasmic reticulum within a muscle fiber releases calcium ions. These ions bind to a protein complex, causing it to shift and expose the myosin binding sites. The myosin head then binds to actin and the cross bridge is established. In the next step, ADP and phosphate are released. Their energy powers the movement of the myosin head. This step is called the power stroke. Actin moves toward the center of the sarcomere. ATP then binds again to myosin and it detaches from actin. ATP breaks down into ADP and phosphate, and the myosin head cocks back. Myosin heads continue to deactivate and reactivate. This causes the myosin filaments to pull together and slide across one another, contracting the muscle.
Okay, so we get an idea here that there's these steps we're going to go through in contraction, and that clip really gives a good perspective of what each of those steps look like. So I encourage you, the great thing about having this lecture on video, you can go back, rewind it again. If you can know those steps, then you can understand muscle contraction no problem. And so again, as I mentioned in class, you want to be able to go through step by step what each of these are. You want to be able to draw a picture of sarcomere, the myosin, the actin, how those overlap, understand the role of calcium, binding troponin, tropomyosin moving, myosin head binding to actin, the power stroke right here, and then ultimately that leading to the muscle cell relaxing. And then the next step of this, which the video doesn't necessarily go into, is that this isn't just a one-time thing. And we all get this because our muscles contract. We don't just have them contract for a millisecond. They often contract for seconds, sometimes minutes at a time. And so this process is a continuing process of, from calcium to troponin, tropomyosin, all that occurring, continued contraction, continued power strokes. We'll get to another lecture, look at, well, that can't last forever, can it? Well, no. You need ATP in order to make this continue to go on, and that's limited. We'll see in the next lecture. But I want us to grasp that one knows what the steps are, know how this occurs, be able to go through step by step how the muscle cell contracts. In our next lecture, we're going to look at how we form ATP from the muscle cells and how certain types of muscle cells are different. We call those muscle fiber muscle cell types, type 1, type 2. We'll get into what those are and why that's important.